So what we're looking at here is the biosynthetic pathway of a polyketide. And this pathway is essentially a multi-enzyme factory which sequentially extends carbon chains and makes polyketides. A polyketide is a complex molecule that is the precursor for many of the antibiotics we're actually using today. What we see here is that this factory is divided into modules, and each module contains several enzymatic domains. One of them is this little orange domain here, which is called the acyl carrier protein, that transfers the intermediates through the different enzymatic domains and also from one module to the next. So what we see here is the cryo-EM based molecular model of the fifth module in this pathway, and shown in blue is the ketosynthase, in green is the acyl transferase, the ketoreductase is shown in purple, and the acyl carrier protein is shown in orange. The role of the acyl carrier protein is to transfer building blocks from the acyl transferase domain, the green one here, to the ketosynthase, and also intermediates from the ketosynthase to the ketoreductase, and finally to the ketosynthase of the next module. The following movie illustrates a recapitulation of the enzymatic cycle as captured by cryo-EM reconstructions. In the first step, the acyl carrier protein of the upstream module, here shown in red, transfers the intermediate chain to the active site entrance of the KS. The acyl carrier protein of this module will then go to the acyl transferase domain to grab a building block, a transition which is associated with a dramatic rotation of the ketoreductase domains. The acyl carrier protein will then deliver the building block to the ketosynthase active site using a bottom entrance. Once the ketosynthase domain forms the product, the acyl carrier protein will pull it from the bottom and transfer it to the active site of the ketoreductase in purple, which will then modify it even further. After this final processing step, the acyl carrier protein will be ejected from the reaction chamber, carrying the resulting product towards the ketosynthase of the next and final module 6 in the picromycin biosynthetic pathway. And so now we have a much better understanding of how the acyl carrier protein transfers all these little molecules from one module to the next and between domains within the same module. And we're planning on using this knowledge to potentially redesign these machines to create new, novel molecules that might have pharmaceutical application.